In this video, we will discuss about this PARP-14 helps the pancreatic cancer cells in proliferation and also helps them to get resistance against this gemcitabine via NF-kappa beta signaling. So we will discuss this how they come up to this conclusions through the experimental results that are presented in this figure. So in order to validate this hypothesis, what they did is they overexpress the PARP-14 and they did the stable overexpression and validate that using Western blot and the quantitative PCR results. And here we can see that PARP-14 is overexpressed compared to the control. And when they check the cells proliferations, they are able to see that in the case of this PARP-14 and the control, that is PARP-14 plus DMSO, they see the high cell proliferations compared to the case where the normal control vector is used, or this PARP-14 with NF-kappa beta inhibitor is used. They can see the significant decrease in the proliferations rate. So this indicates this points out towards the fact that NF-kappa beta might be playing a role in conjugation with PRP upregulations to get pancreatic cancer cells high proliferations rate. And these things they have also validated using the colony formations. Here we can see that compared to the control, the overexpression of PARP-14 gives more colonies, and the control, that is PARP-14 plus DMSO, also gives the more colonies, but PARP-14 with NF-kappa beta inhibitors gives the less colonies. And these things are quantified on the right hand side, where we can see that the NF-kappa beta inhibitors lead to the decrease in the number of colonies. And when they check the apoptosis in these different cases, what they are able to see that in the vector they see some apoptosis or apoptotic cells, that is around 5% and 1.85%, and these things goes down as with the upregulations of the PARP-14. It can be seen that apoptotic of the cells goes down from 1.85 to 0.82 and 5.05 to 2.62, and these things remains almost at the same level with the PARP plus DMSO, that is controlled. However, when they use PARP plus NF-kappa beta inhibitors, they see an increase in the apoptotic cells. That is, it increased from 5.05 to 10.98 and 1.85 to 2.93. Or we can see that it negates the suppressions done by overexpression of PARP-14 in terms of the apoptotic cells. And these things are quantified on the right hand side, where we can see that compared to the vector, we see a less apoptotic rate in the PARP-14 overexpressions and PARP-14 overexpression plus DMSO. But when this NF-kappa inhibitors is also introduced in the PARP-14, we see a high apoptosis rate, indicating that NF-kappa beta might helps in the survival of the pancreatic cancer cells in conjunction with PARP-14 overexpressions. And after that, they check the survival rate using the gemcitabine treatment in those different conditions, and they are able to see that when they use PARP-14 and NF-kappa beta inhibitors, the proliferation rate goes down with increase in the gemcitabine treatment concentrations, and it is incomparable to the vector, that is control case. Whereas in the case where PARP-14 is overexpressed and is controlled, that is PARP-14 plus DMSO, they see a less decrease in the proliferation rate. So this indicates that is the NF-kappa beta inhibitors helps the pancreatic cancer cells in survival when and its functions got accelerated in the case where PARP-14 is also overexpressed. And when they check the IC50 value, that is the quantifications of panel E to panel F, they are able to see that IC50 value for the high expression of the PARP is high compared to the case where high expression of PARP-14 plus NF-kappa beta inhibitor is used. So this all pointed out to a fact that the PARP-14 helps NF-kappa beta or enhance the NF-kappa beta signaling, and that in turn helps the pancreatic cancer cells to acquire the proliferation characteristic, at the same time give them or provide them resistance against the drugs such as gemcitabine. By suppressing this NF-kappa beta signaling, we can make the cells make the cells sensitive towards gemcitabine, at the same time also reduce the proliferations of the cells. I hope you understood the final conclusions that comes from this paper. And if you need to understand more about these experiments and the paper in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.